Hi, I'm Dr. Brian Kaufman. I'm a family doctor with CLL. I'm here at ASH 2014 in San Francisco. Dr. Kipps.
I'm Dr. Tom Kipps. I'm at the University of California in San Diego at the Moores Cancer Center. And so I'm very happy to join you today.
And, Dr. Kipps has been my doctor since I was diagnosed 9 years ago. And one of the areas, there weren't any papers that I saw here particularly, but one of the areas of research that we've talked about before is ROR1, and you've got an open trial now, and can you bring us up to date on what you've been finding out and tell us a little bit about that trial?
We're very excited because this is at the early stages, but we've manufactured enough of the antibody to, under conditions that would be required to introduce it into patients. And we've also gone through a number of important regulatory tests, including safety testing, and we've looked at the antibody both in other animals and in primates. And this was all necessary for us to file what's called an investigational new drug designation, which we filed this summer with the FDA. And so we're very excited in July to finally receive approval to go forward with clinical trials. And so clinical trials now have been launched at the Moores Cancer Center. And
that's at UCSD, yeah.
At UCSD. So we've now, I believe 3 patients have now been treated, and so far we're very excited. There have been no serious events, no actually adverse events to speak of, and that's very important to us.
Right.
And we're seeing some early signs which are encouraging to us in terms of hitting the target, and I think this is quite exciting. Obviously the ROR1 protein is something that we've identified as being on CLL cells, but it seems to play an important role in how cancer metastasizes and how it regenerates itself. And so by attacking this protein, we're hoping that we might get at some of the features that we'd like to wipe out in cancer. Of course, the exciting discovery also of the ROR1 being on not just CLL, but many other types of cancers. And this is something that may mean that the work in CLL, if we develop the antibody, could actually have an impact in patients with very solid tumor malignancies. And more recently, work that has been done with members in the laboratory have actually identified it on a cancer stem cell. That work was just published a few weeks ago.
So tell me about the cancer stem cell, a little bit about the theory behind that.
Well, this is the theory, and obviously there's debate on a lot of issues, but I think what the stem cell hypothesis is that the cells within a cancer are not uniform. And there's now evidence for this. And you may have cells within the cancer that actually have a regenerative capacity. They are the stem cells. They are like the seeds of the cancer. These cells tend to be growing more slowly, and they tend to be more resistant to the drugs that we use to treat cancer. And they may account then for why patients under therapy may have a response because it may kill off the other non-stem cells, and the stem cells are still present in small quantities. These cells may have the opportunity then to spread to other parts of the body as well as to grow back after treatment. They may account then for relapse. One of the perplexing things we're seeing in CLL is why does the disease come back? Another question that we're addressing today is why is there persistent leukemia in patients treated with this very successful drug, ibrutinib, in that you're seeing, especially early on, mostly partial remissions, and what accounts for why these cells are still present there? Why doesn't it wipe out the disease entirely? Now, of course, time will tell. I know that there's some work to try and define whether over time maybe there could be, these cells further being reduced over time. But I think it's fair to say that if we take up this hypothesis that there may be a collection of cells which have unique qualities and that may be more resistant toward therapies, we have to devise strategies to go after them, because they may not account for the bulk of the disease, but they may account for the parts of the disease that account for relapse and account for the cancer spread. So this is something that we have to think outside the box. Now, finding this protein ROR1 on leukemia cells, but also on these other types of stem cells, I think is exciting, because we might be able then to address the stem cell and extinguish the cancer at its roots, which I'm very excited about. I'd very much like to have CLL cured. I would like not to consider it a disease that we treat like hypertension. I'd like to see if we can actually have CLL and have therapies for patients which are safe, that are effective, and are allowing patients to get off therapy and be able to lead normal lives as if they had maybe a pneumonia and they've gotten over the pneumonia. Now they can go on with their lives without having to worry about relapse disease. This would be very exciting. And I think, having been familiar with this disease for many years, this is certainly something that I think would be the goal that we should have. And not just to be complacent at being able to manage or get by.
Right. So what I'm liking about this is you're taking a drug that was developed, or an antibody that was developed for CLL, and saying, hey, it might have applications in other places. And we're kind of seeing also the opposite happening. The PD-1 blockade, the checkpoint blockades are now being looked at in hematological malignancies. So there seems to be, the hematologists and oncologists are talking to each other a little bit more.
Well, I think that it's actually gone more in terms of the direction of blood cancers to solid tumor cancers. I mean, many of the drugs, a great number of the drugs that are being used in solid tumors and treatment of patients with cancer in general cut their teeth in the therapy of patients with blood cancers. We see that with monoclonal antibody therapies, first approved for patients with leukemia and lymphoma. We see it with very small molecules that are having unique targeted effects in cancer. So this is not a new thing. What is new is that I think this PD-1 that you mentioned was developed with the idea of how do we take down the immune system so that we can actually prop it up to be able to fight the cancer. And actually the data would suggest that this would be very, very good to do in blood cancers. Unfortunately, I think some of the pharmaceutical companies want to target the larger number of patients that have solid tumor cancers. So it's been a struggle to get it tested in blood cancers, but I do have a lot of excitement for those approaches as well. I mean, obviously, if we can activate the immune system to go after the cancer cells, that's great, because that may also be a measure of trying to cut down the risk of relapse. But if we have also different modalities of treatment that don't work through the same mechanism, then we might also be able to kill the cells that we are concerned about as accounting for relapse. Now with the ROR1, we have it early going, and I must say that our major concern is how well patients are doing, and they seem to be doing well, and one patient actually feels like he is improving, and I think some of the data that we have is very exciting. And we're starting out with low doses, but I do think that this is something that I'm even more optimistic now than I was last year at this time, because we actually have the antibody now in patients, and we're seeing very encouraging things.
So if patients are interested in this, they could go to clinicaltrials.gov and put in CLL and ROR1, and your trial would pop up.
And I'm hopeful that we can get this antibody used to maybe it would be a great drug to use in combination with agents such as ibrutinib, and be a 1-2 punch that we can use to wipe out the disease, and hopefully allow patients even to consider getting off therapy. That would be very exciting.
Yeah, well, I'm with you 100%. I mean, I think there's not a patient out there that wouldn't prefer a cure to a control. So I'm very happy with the control right now. That control is way better than uncontrolled. So let's take a quick break here, and we'll pick it up in a minute.
Okay.